Welcome to this explainer. Today, we're diving straight into the ultimate micro-mystery, tuberculosis. We're going to treat this notorious pathogen like a stealthy, calculating invader, and we'll be stepping right into the shoes of our clinical lab scientists, the forensic detectives whose job it is to track it down. We've got a genuinely fascinating biological heist to decode today, so let's get right into it. So, here's the big question driving our investigation today. How exactly does this microscopic invader break into the highly defended fortress of the human respiratory system? And more importantly, how do our laboratory detectives catch it in the act before it causes irreversible damage? This is a pathogen that has successfully outsmarted human medicine for millennia, adapting to our defenses at literally every turn. To understand how to beat it, we need a dual focus. We've got to figure out the intricate biological crime happening inside the lungs, and then we have to master the forensic investigation required to actually stop it. First up, let's look at our primary suspect, Mycobacterium tuberculosis. It's an airborne bacterial pathogen, but let's be clear, this is not just your standard bacteria. What makes this specific organism so exceptionally resilient is its biological armor. It has this uniquely thick, waxy cell wall that's just packed with mycolic acid. And this armor, it's the ultimate secret weapon. It allows the bacteria to survive suspended in the air for long periods, and it fiercely resists our body's initial immune attacks. When an infected person coughs or even just speaks, they release these droplets into the air, creating the perfect opportunistic vehicle, a tiny, invisible droplet carrying an armored invader, perfectly equipped to find a new host. Part 1. The Invasion, Pathogenesis of Tuberculosis. If we track this invader's precise timeline, it is honestly a masterclass in biological infiltration. At minute 1, the host takes a breath, and those airborne droplets are inhaled deep down into the tiny alveolar sacs of the lungs. Fast forward to hour 24. The bacteria aren't just floating around anymore, they've actively anchored themselves to the lung tissue. But by week 2, the plot thickens. Our immune system recognizes the breach and sends out its cellular guards, the alveolar macrophages, to essentially eat and destroy the invaders. But here's the crazy part. Mycobacterium tuberculosis actually wants to be eaten. Once it's inside the macrophage, it deploys these advanced immune evasion tactics. It literally hijacks the host's defense cell, stopping it from destroying the bacteria, and instead uses it as a safe house to survive and replicate. By month 1, this silent buildup triggers the formation of granulomas. The immune system, getting desperate, tries to wall off the bacteria in a cellular prison. It's a remarkably stealthy timeline that completely turns our own defenses against us. And the ultimate consequence of this prolonged standoff, it's a catastrophic physical transformation within the lungs. On one side, you have healthy tissue, normal function, beautifully clear airways. But on the other side, you have infected tissue being ravished by a massive inflammatory response. As that standoff inside the granulomas breaks down, the pathogen triggers a massive cytokine storm. The immune system essentially goes into overdrive, leading to caseous necrosis. Now, that's a clinical term for the lung tissue basically decaying into this cheese-like substance. Eventually, this dead tissue liquefies and creates empty, hollowed-out spaces in the lungs known as cavitation. So the key takeaway here is that it's actually the host's own aggressive immune response desperately trying to clear the bacteria that physically destroys the lung architecture. This severe structural damage is exactly why patients with advanced TB experience that terrifying symptom of coughing up blood. Moving on to Part 2, the investigation, step-by-step laboratory diagnosis. Okay, let's shift our perspective from the biological crime scene straight into the forensic laboratory. When a specimen arrives, scientists follow a highly specialized sequence to draw this elusive pathogen out of hiding. It kicks off with specimen collection. Because we're dealing with a dangerous airborne pathogen, scientists have to process deep lung sputum samples inside specialized negative pressure rooms, just to keep the whole facility safe. Then we move to microscopy. Remember that waxy mycolic acid cell wall we talked about? Well, because of that armor, normal stains just won't work. Scientists have to use specialized acid-fast bacilli, or AFB staining, to reveal the bright red rod-shaped fingerprint of the bacteria. Following that is the culture phase, where the sample is put into specific environments like solid Lowenstein-Jensen agar to coax the bacteria into growing. Finally, we bring in the heavy hitters, molecular diagnosis. Labs use cutting-edge biotechnology to run rapid PCR tests, amplifying the pathogen's DNA to confirm its identity beyond a shadow of a doubt. Now, what's really fascinating about this workflow is the high-stakes clinical balancing act our lab detectives have to perform. Because this organism hides inside cells and grows incredibly slowly, diagnosticians have to weigh the pros and cons of our major diagnostic tools. Bacterial culture is still the absolute gold standard. Why? Because it proves the bacteria are actually alive. Having a live culture is the only way to test if your prescribed antibiotics are actually going to work against a potential multi-drug resistant strain. The catch? It is agonizingly slow. It can take weeks to grow. PCR, on the other hand, offers a massive advantage: speed. It detects genetic material in a matter of hours, letting doctors isolate an infectious patient immediately. But detecting DNA doesn't always prove the bacteria are currently alive. So the most accurate strategy? Use the rapid molecular intel to act quickly, but rely on the slow, meticulous culture to secure that final actionable conviction. Which brings us to part 3, the verdict. Key takeaways. Let's close out this case file and consolidate the absolute most important facts. 1. Our primary culprit is Mycobacterium tuberculosis, heavily armored by its mycolic acid cell wall. 2. Its entire MO relies on advanced immune evasion, specifically hijacking alveolar macrophages for its own survival, which ultimately provokes our immune system into destroying our own lung tissue. And 3. On the investigative side, despite the sheer brilliance and speed of molecular PCR testing, the meticulous process of bacterial culture remains our ultimate gold standard for detection. So, I'll leave you with this final thought to ponder. As these microscopic invaders continuously adapt, literally evolving more and more complex ways to escape our immune systems, what kind of incredible new tools are our laboratory detectives going to have to invent next just to stay one step ahead? The biological heist is always evolving, which means our forensic science has to evolve right alongside it. Thank you so much for joining me in this explainer. Keep questioning, keep learning, and I'll catch you next time.